Hi, everybody. It's really exciting to be here. Thank you, the organisers. I'm delighted to have this opportunity. I hope to entertain you, if nothing else, and to bring, as the Provost said earlier, perhaps a different perspective. So I'm a senior lecturer at York. I suspect we could fit York onto your campus. This is absolutely enormous, your space here. But we're very old, and we have a big university to the south with about 20,000 students that come there to learn, and I work within the department there. You can see some students and boffins working away on the slides. So my expertise that I bring is synthetic biology-based approaches, engineering biology, as a way to help with the critical mineral issues that we're approaching. I also work on bioinformatics and plant-based metal recovery, particularly focusing on the critical minerals of nickel, platinum group metals, and rare earth elements. And I'll give you little snapshots of some of the work that we're doing towards that. I also have a side hustle in organic and inorganic pollutant remediation, explosives, heavy metals, which is often a combining effect when you're looking at recovering metals from more dilute areas of contamination. We work with biomass crops, real plants, as well as model species, so willow, miscanthus, and eucalyptus. And we work with industrial partners very much to understand what it is they want, rather than working as an academic silo. As Joel said, I'm a co-theme lead for food, water, and waste at the York Environmental Sustainability Institute, and that gives me access to a lot of wastes, environmental wastes and sources, so I'm known as Dr. Waste or Dr. Poo, but we work on all sorts of waste. So we've got some pictures there of rare earth element mine sites on your top left. We've got explosive sites. In the middle there's a little gold mining site, sweeper waste, commercial waste, and rare earths in things like coal fly ash and other waste sources, where we're looking at removing relatively low amounts, low concentrations, but high volume wastes. I'm also a co-theme lead. I'm a work package lead for Elemental, the Engineering Biology Mission Hub. Elemental is nice and short. Our actual title is quite wordy. We're the Engineering Biology Hub for environmental processing of metals and contaminated land to industrial biotechnology and a circular economy. I think we just tried to get all the words that would tick the boxes that got the funding. And so there's a number of us across the UK working together from these different universities. My work package focuses on plants, but the rest focus on microbial-based remediation and recovery. And so I'm very, very happy, delighted to talk. If anybody wants to know more about that, just come and ask me. Here's a good picture of the team, lots of mugshots of smiling, happy British academics that want to come and work with you. So again, let me know if you see anyone up there that you'd like me to introduce you to. I also work on the social science, so I work with social scientists because my work is transgenics, GMO, and to push some of that, particularly coming from a European side, is something that we need to work on as academics. We can't just work on our technology and expect it to be ready to go. There are big reasons why we can't push some of our GM technology into Europe at the moment, although there are big changes. So earlier this year, the EU approved the new genomics technique proposal, and things are moving apace there as well. So I think this is really, really promising area, and I'm very happy to devote some of my time to this social science facet to help my technology ultimately move forward. So what are technology critical metals? I really hesitated before I put this slide in because I thought pretty much everybody in the room would know, but I'm the first person to speak, and I think it would be very remiss of me if somebody in the audience still didn't understand quite what this was. So just put it on hold otherwise. They're high adverse environmental impacts per kilogram of metal, so they're really, really difficult to mine. They're geopolitically highly concentrated, so you guys have got a lot, China, Russia have got a lot, some other continents. Europe, we've got nothing. Help. Increasing demand in emerging technologies. I can see all the smartphones and laptops. We all need these kind of things. And a growing number of countries now maintain official critical mineral lists. So I found out a couple of days ago that New Zealand, the pristine land, now have a critical mineral list. So everybody is scrabbling for these metals. Some of the reserves are running out. I know we've got plenty of some, but nickel, for example, at current supply, 35 to 40 years left of supplies that we can mine using current techniques. So it's time to bring in innovative new ways to eke out the remaining reserves that are at lower grade, lower concentrations. And some of these, the next few slides are in the International Energy Agency's Global Critical Minerals report that's just come out. And you can see the increase in prices of all these metals that are critical metals that we're all really, really scrabbling for. So you've got your magnetic rare earths, your battery materials, and the prices there, you know, they're doubling over since the last year. So things are really, really ramping up. And you can see that the growth, some of this is from a growth in renewables. So we've got the solar panels, wind, electric cars, and battery storage, and the demand for those critical minerals is being driven by the increase in production of these climate-tackling technologies. So we're playing a big role here. We really need to provide these metals, these elements, as quickly as we can. Venture capital is also spending really, really highly in this area now as well. There's a little up and down a bit, but basically there's a lot of money coming in from venture capitalists to support our academic activities and help that go out into new startups and into existing industry as well. So this is really, really encouraging. Alongside that, there's plenty of money around to do this. It's not something I'm used to saying. We have money to do this. That is, I'm here today because of the NSF funding, and you know, this is something that we've got time and, well, maybe not so much time, but we've got money, resources, and expertise in the room to really address this. So what's happening to those metals that are already out there? The recycling is increasing, but it's still a long way to go, and I found this really quite dramatic. For these different metals, these different critical minerals, our recycling is woefully inadequate when these are so, so valuable to us. We're really not turning these back round. So this is an area that we really, really need to invest in. And I'm going to talk about how plants can be used for this, because a lot of this recycling is from metals that we've already dispersed back into the environment, so legacy waste, mine tailings, vehicle catalytic converters, for example. There's a little graph there, top left, on the palladium, which is used predominantly in catalytic converters, which we do recycle from the vehicle, but at that point, 50% of the platinum group metals have gone out of the exhaust across the roads of the world. I can't think of a better way to dilute something into a place that you can it's very hard to recover from. So my interest particularly is using plants to recover metals from very dilute wastes. And this pollution that's caused by mining is a big, big issue. So I can see that we're very, very concerned. This is the BBC News here, and we're highly concerned about Keith Richards' arthritis. That's very, very concerning too. But 23 million people worldwide are affected by metals. Maybe they're more worried about Keith. And we are poisoning ourselves. This is a really poignant case study here in Ghana from Gold Rush, which artisanal mining for gold, little pockets that collectively add up to a huge environmental disaster. How do we go in and clean that when we can't even access the site? What can we do? How can we recover that and clean up this mess? So it's a huge, huge problem. I think this is also a really interesting slide as well that came out recently in Science last year, showing the metals that are contaminating cropland across the world. And we think, well, maybe I think I'm safe in Britain eating my lettuce, but I'm also eating my mung beans from China and maybe my corn from the US or wherever it's being grown in the world. It's a global problem. You think you're pristine and clean wherever you are, but we all live on the same planet. So can we use plants to recover metals? And if we did, what would that do? So as I've said, we could recover from more dilute sites that are commercially unviable using existing mining methodologies. We've got environmental restoration, so you might want to go out there for a family holiday with your canoe and go fishing. I wouldn't recommend it right now. This is a site in the US, in Vancouver, a palladium mine that's active, so it's not revegetated because it's still working. But once they've finished revegetating, this is going to be a challenge because there will be metals in that soil that will stop the plants growing. We've also got phytostabilization that we can offer. There's a tailing dam in the foreground there which breached a few years ago, and the waste went into the river, causing environmental damage. If we can get plants that will grow on these areas, maybe we can phytostabilize. We've got metal recovery, of course, what we're after, maybe dilute, but very large volumes. Can we use that to recover these metals? And if we do, are these metals worth a bit more? These are eco-metals that have been gently plucked from the soil using plants. Should there be a premium there for that kind of approach? Would Elon Musk be interested? I'm listening, I'm ready. But these metals are at phytotoxic levels, so what can we do? as biologists, that's the problem that I want to tackle. And this is my favorite slide because this is a picture of plants that have evolved naturally the ability to hyperaccumulate very, very high levels of metals from geologically rich areas over millennia. So in the center is my favorite picture of Pycnandra, which is a tropical tree which grows in Sabah, Malaysia. And it's a massive tree, but there's about 12 kilograms of nickel in that one tree. And the sap that you can see coming out is 40% nickel. So if you know your chemistry, that nice dark kind of turquoisey blue, that's the nickel that's in there. So there's biology there, and there's selectivity because that's just nickel. And the other plants there are solely zinc or arsenic or lead, thallium. So there are plants out there with that biology already existing. But you can see from these plants that they're quite small. Some of them are growing tropical. I'm not going to grow a tropical tree in Yorkshire. It's not going to work. So we need to transfer this biology. We need to understand it and move it into systems that we could work with. So how far is engineering biology getting? Well, it's very, very much on the periphery, but there are a few companies now that are using engineering biology to help with metal recovery. So I've listed the ones that I could find. Most of them using bacterial consortia, so they're not necessarily actually using engineering biology per se, they're just using naturally found organisms to help. But some of them are also using CRISPR-Cas or other engineering approaches to modify them. And the bottom two there, botanical and geomines, are using these hyperaccumulator plants and looking at ways that they could engineer those plants too. So we are on the cusp of developing this technology for field applications. But one of the problems here is that there are no hyperaccumulators of platinum group metals and gold, so these are things that we want to recover as part of our technology critical metals. And if you look at this graph here, you can see on the x-axis, you can see the threshold for hyperaccumulation. So if you move to your right, you can see there are lots of metals on the far right that plants hyperaccumulate very, very highly. So manganese is right over on the far side. And then if you look on the y-axis, you can see the value. And so you want something that's kind of far to the right and quite high up. I've highlighted nickel there in green, and you can see this is actually phyto-mined at the moment. This is the cherry picking that we're pulling off there. It's worth a lot. There are plant species that will hyperaccumulate it. So that's really, really good. But then if you look, well, what else could we pick off? There's thallium, cobalt, and manganese look like the next ones, and there is some work going towards that. But as you work your way across, you can see the platinum group metals, they're not naturally hyperaccumulated, which makes complete sense because there's been no evolutionary pressure. We don't have areas that are rich in gold that plants would grow on and develop that. And the chemistry of these elements mean that they're not available biologically for plants to take up. Rare earths there, you can see there's lots of rare earths that are hyperaccumulated, and we'll go into that a little bit later to discuss. So my job is to engineer plants to recover technology critical metals and remediate environmental pollution, to put these two alongside each other. So this is my favorite Rumpelstiltskin story. Well, my favorite one was the enormous turnip, but Rumpelstiltskin was second. We want to take this almost literally plant biomass straw and weave it into gold. That's what we'd like to do. We'll all be rich. And this is my approach, which is a four-pronged approach here. We start with solubilization in the soil. How can we get those metals taken up by the plants? How do the plants then take it up and mobilize it around the plant? What happens once it's inside, because it's going to be toxic? And how are the plants going to store that? Whereabouts are they going to store it? It's no good if it's in the leaves, if they drop off in the autumn. We want it in the stem, and we can use biology to work on all of these different factors. So the first step, solubilization. I've mentioned before, gold, palladium, and nickel, they exist in zero-valent form. So this is kind of a nugget that Hank and Marv found on the range, and it was exactly as it was for millennia. It's geologically, it's inert, and it's unchanged. But for plants to be able to take that gold up, it needs to be as a cation, it needs to be solubilized. And cyanide is used to do that extensively in the mining industry, to solubilize these metals. So some plants and some rhizospheric soil bacteria exude cyanide. And so we've started to look at that as a way that we could approach this. So here's a relatively simple graph explaining this. So a lot of plants are cyanogenic. Here we've got Lotus japonicus, which produces cyanide. We think as an anti-herbivory it would stop things eating it. And we have a mutant that lacks the ability to make cyanide. So if we grow these plants on a soil without nickel, which can be solubilized by cyanide, we can see that the mutant grows slightly less well, and that probably because it's using that cyanide to solubilize some of the metals that it wants, or that anti-herbivory. It gives a little bit of a benefit to the plant at that point. But when we grow the plants on soil that contain nickel, we see that the mutant grows better than the wild type. And what's happening there is the wild type is solubilizing that nickel at very high levels, admittedly, and it's killing it. It's taking up high levels, and the lower graph shows that the leaf nickel content, you can see the wild type has got a lot more nickel in it. So this works in practice. So what we're looking at is engineering that cyanogenic pathway into plants, and it's a relatively simple pathway, and we've got this working now. You can see that several gene products will make this a cyanogenic compound here, which can then be cleaved. So as the roots penetrate through the soil, they get damaged, and a cyanide can be released, and you also have root herbivory, and so you're just producing the cyanide here. We've got it in front of a root hair-specific promoter, so we're not making cyanide throughout the plant. We're making micromolar levels of cyanide that will just solubilize right next to the root. So if the root is near the metal, it will solubilize it and take it up, but it won't touch anything that it can't take up. So we've got some preliminary data showing that we've got expression in the roots, and we've measured cyanide being produced there, and now the next step is to test these in soil and see if that's going to give us the same results with the uptake of nickel, and we'll try other metals too. So the next step is that the translocation of metals, and we've done some work on transporters, the kind of molecular gateways into the plant, if you like, and there are lots of different transporters out there. I'd say something like 10% of the Arabidopsis plant genome is transporters, so lots and lots of different genes. We've done some work here, some PhD students I see there showed that this copper transporter will also take up gold and palladium, and so there are ways that plants can take up critical metals that they don't have any need themselves for. Okay, they need gold, but they need copper, but they don't need gold and palladium, but we can get them to take that up. There's also a rare earth element transporter that we've characterized, and so we had some visitors there that came that I worked on with this paper. That's my house in the background in the UK. And we, thank you, we all live like that. And so it's a rare earth element transporter. And I think this is a really interesting slide because this is a Dicronopterus linearis is a tropical fern, and it grows and hyperaccumulates rare earth elements. And if you look at the top figure there, you can see on 1 micromolar neodymium, maybe 10 micromolar, the biomass actually increases. And the Chinese have known this since the 1970s, that rare earth elements will increase plant biomass at really low concentrations. So they've been actively adding it to their agricultural soils since then. Less so now because they can see that there are some downstream problems, almost to the point where this is now a problem for them, that they need to kind of remediate and reduce these levels. So that's an interesting aside. What these rare earth elements might be doing in biology is also fascinating to me, but less interesting for the mining side of it. We've overexpressed this rare earth element transporter, and you can see in the middle panel here that it gives a corresponding increase in the ability of the plant to take up rare earth elements. So there's a degree there where we can tweak the biology to uptake the elements that we're interested in. On top of that, separation is key. Oops, I'm not sure where that's gone. There we go. So back to this graph that I showed before, and you can see that the rare earth elements are all on top of each other, but some of them are a lot more valuable than others. So terbium at the top, cerium nobody wants, you can't give it away. So a little picture here that kind of expresses what it's like in a chemical form. It's like these Bobby and Kenny's sextuplets. They're almost chemically identical. How you separate them is very, very difficult, environmentally damaging. It needs a lot of acid and other treatments. Can we use biology to separate these? And to a degree, we already can separate some of these. So we're investigating ways that we can bind specifically some of the rare earth elements over other rare elements. If we can do that in plants, and then we can get the plants to take up the ones that we're interested in, then that has got great value looking forward. So that's a really important area of research that we're really focusing on. So engineering that rare earth selectivity in plants and using biology-based methods is I think, really, really promising technology. There's lots of different rare earth rich wastes that are out there. We've got coal fly ash, we've got mine tailing waste as well. But some of our studies have also suggested, proven that we need a very low pH to get the rare earths taken up by plants. And we're looking at engineering the rhizosphere, so plants produce acidic exudates, and so do some bacteria. But we also sometimes have mine wastes that are very acidic, or acid mine drainage, for example, that might be able to co-combine to help release these elements for uptake by the plants. We're also looking at nickel phytomining and trying to identify ways that we can harvest that biology of those nickel hyperaccumulators that are already being commercially farmed and improve that more. And so we've got a diagram here explaining how nickel and organic pollutant detoxification could be tackled in plants. So I've been out to look at nickel hyperaccumulation in Sabah, Malaysia, where it's geologically really rich, and I've been out a few times. It's great that all the plants there, they grow on these really high levels of nickel, and there are plants in the Siliciaceae, which is the willow family, which is one of the biomass crops that we work on at temperate species. So we want to grow a temperate biomass crop that could take up nickel. What could we learn from these tropical willow cousins, if you like? And so we've been out there working with these people in Sabah to understand that biology and bring some of these plants back and study the transcriptomics. Alongside that, we've moved more work into the temperate willows to understand what's going on in their responses to the nickel. And willow is a great example to work with. It's really, really diverse genetically. We've got a really big collection in the UK. And it grows very, very well. There are lots of studies. It's been used for phytoremediation. It's a native species. It grows in all sorts of different niches. It's great. We really love willow. So what we did was we took about 20 different species and we screened them on soil for nickel resistance to see if we could get ones that would spread and which ones were most tolerant and which ones were most sensitive. And what we found was it split into two camps. So here you can see we have nickel-sensitive willow lines and we've got nickel-tolerant lines. The sensitive lines we found out when they were grown without nickel, they grew very, very well. These are all the biomass lines. So they've got these wonderful British Empire names. We're very proud. We've called them Resolution, Endurance, and Terra Nova. And they grow like absolute stink, which is great. But on the other hand, the nickel-tolerant willow lines, you can see over here, these are more traditional ornamental varieties like White Welsh and Lambertino and Caliantha. So very, very traditional old lines. They are less affected by the nickel. And we were not expecting to see this big split. So it was very interesting. But I think a picture speaks 1,000 words. And so if you can see that the biomass was slightly more decreased in the nickel-sensitive willow lines, actually they're dead. This clearly shows that they could not survive the levels of nickel that we were giving them. Whereas these more traditional varieties are barely affected, I would say. They're still green and they're still flourishing. So we then did some transcriptomics. We wanted to see what genes are evolved and responsible for that. But we also wanted to measure the levels of nickel in these plants so that we could understand more. And when we did that, we found that all these biomass lines that were growing like stink were taking up the nickel. And that's what's killing them. They're absolutely full of nickel, whereas the traditional varieties are excluding that nickel. Okay, they're growing more slowly, but that metabolic cost of controlling their uptake of metals and their transporters means that they didn't actually take up the nickel in the first place, they sensed it. So that's really interesting, but what we're interested in here is these biomass lines, they're taking up nickel, that's great. Okay, they're dying, which is less great, but that's the next step. How can we make them survive that? And how can we then, another step towards a hyperaccumulator? So the next step in our theory here is metal binding proteins. Are there ways that we can collate these metals? And so we're looking at pathways that will produce nickel, or in this case, we've got palladium binding proteins that will bind or collate and store those metals away from the biological machinery in the cells, so the plant can continue to take up these metals without popping its clogs. Okay, so a couple of pictures there. So we've got a binding protein, azurin, that binds copper, and it's a beautiful blue color. And we've got a mutant in that binding protein that will bind palladium. And the copper gives it that lovely blue color, but when it binds palladium, it looks like the River Ouse in York, which is quite brown. That's not great. But that's the kind of biology behind it. The final step is nanoparticle formation or slash localization. So you don't want to put your metal into the leaves and then in the autumn that all just goes back onto the ground. You want to partition it into an organ within the plant that you can harvest, so the stem, for example. But platinum group metals and gold, they also, when plants take them up, they make them into nanoparticles. And we've got ways to affect this nanoparticle formation within the plant. So we've engineered the size of gold nanoparticles within plants using these short-chain peptides. So these are short amino acid sequences that help atoms of gold out there become reduced and bind and form nanoparticles. And by expressing these in plants, we've shown that we can produce a range of different nanoparticle sizes. And the chemists get very excited about this because the smaller the nanoparticle, if you can keep it small, more and small rather than big and not so many, you've got much higher catalytic activity and catalytic reuse time as well. So that means that you've got a downstream product that might have more value. So that's, I think, a really exciting area as well. So not just taking up the metal, but thinking of what form you could put that metal into that will add more value to the plant biomass. So if we put the components together, we've got solubilization, uptake, binding, and storage. We can see the push that we're providing from the base to bring the metals into the plant and the pull at the top to sequester and bind and move those metals into forms that we want. And the value is of a very rich plant biomass, which we could then smelt. So ideally, I'd like to say this is what we'd like to produce, this beautiful, there's always a metal tree sculpture somewhere nearby. I found this placebo in Forest Park by John Payne. I'd like to think that that's a palladium plant that I will be producing in the future. Yeah. And then just finally, one more step, that not only could we produce metal-rich plant biomass, but we could add more value to that. So metal-rich, particularly for platinum group metals and gold, and nickel too, these are catalytically active metals. When they're biologically bound, can we use them as a catalyst? Can we use that catalytic activity either as a catalyst for commercial chemical reactions or to get out the rest of the value? I'm searching now to see any wood in here, but maybe I'll look out for a tree. So wood is maybe 40% sugar, and then maybe another 40% lignin and some other components as well. So the sugar, cellulose, we can ferment for biofuels, which we want. The lignin, we can fractionate, like you did in your chemistry classes at school. You remember you drew out the fractionation for crude oil, and you can get your plastics and your lubricants. You can get the same from lignin. It's a complex of different aromatic compounds, and we can fractionate that lignin component and make lots of biopolymers and products that would replace fossil fuels. So to just burn the biomass and recover the gold seems, really quite a step back. This is a step forward. This is the future, adding value to that metal-rich biomass. So almost there, all right, it seems we have a lot of picnics and cakes, and this is very important. Cake just releases so much academics discussion. I've found the sugar really helps. It's very important. I should be putting a new grant in for cake next year. And we come up with ideas, and then we go back to the lab, and it's why I'm here today, because it's just talking face to face. It's a real pleasure to be here and actually kind of talk. I can do Zoom, I can, but this is so much nicer. So it's great to be here, and thank you for listening.
Thanks for a great talk. I had lots of questions, but one is just about thinking about why the plants do this. So for example, for the rare earths, is there a physiological reason they'd want to hyperaccumulate it for the plants, or could it be for microorganisms that live in association with them?
Yeah, no, that's a fascinating question. Thank you, Yashko. Microbes will also interact with rare earths. Plants will take up rare earths. Traditionally, I've had grant proposals refused because people say, oh, rare earths are not important, Liz, why are you looking at that? But we don't know. They're at astonishing dilute levels, but they're everywhere. And there's some very exciting recent research that shows that rare earth elements can replace cofactors in enzymes in bacteria, so an alcohol or aldehyde dehydrogenase, for example, and it would change the kinetic abilities of those enzymes. To me, that's really exciting. So I think that we're in a place a bit more perhaps dogma of this metal is in this enzyme. It's always been copper and it always will be. But what's actually happening are these rare earths replacing as cofactors and changing biological activity and function, and then thus what the plant and the microbe collectively are doing. I didn't talk very much about microbes today, despite the title of my talk, but they're very much alongside, within and around plants at the whole time. Yeah, great, thank you.
Oh,
Yeah.
great talk. I have a couple questions, but I'm going to try to limit it to like one or two. So I saw you showed a graph where you were showing like certain amount of metals, incorporated into these plants. I guess the simple question is, I'm assuming what you're trying to do is like kill two birds with one stone. You're trying to, in this case, may clean up an area that's an environmental disaster while also picking out these metals out of these plants and using it, for something later in the future,
Yeah.
But I guess what I'm curious about is like, what amount of metal accumulated in these plants would be considered like economically viable to us, like, or significant to us? Like I've seen that you showed a graph that showed like some of these plants may take 500 parts, or I think it was parts per million of like nickel or something, but that may not be as relevant to us as like maybe 1,000 or 10,000 or something. So
Yeah.
I guess that I'll just start to leave it at that. And
Yeah, no, that's a crackingly good question. I could see somebody nodding behind you. Yeah, no, you're absolutely right. Concentrations are incredibly important. Do the plants take up enough metal? Is that going to be valuable to mine? Okay, there are different prices for different metals. So perhaps if you're approaching something like palladium or gold, that you don't need quite so much in the plant. You've got the value of remediation, which is increasing. There's value there. There's value in all the having the plants present. It's almost like a bolt-on. If you're growing, you have to revegetate or clean up this waste. You're going to put plants on it. You can harvest those plants and ash and leach, and that's what botanical and geomines do currently. And it's commercially viable, the levels that they're getting out. So it's how much can we increase using biology? We don't know what the upper limit is that we'll be able to get into plants. We're still in the infancy there, but yeah, it's as much as we can.
I guess I was taking notes while you were going through, so there may be a couple parts that I missed out on, but like I guess what I'm thinking about is like you're trying to use these plants to pull out certain metals, and you're trying to do this, and I'm assuming like this is going to be like a long-term thing. This is not something that you can do overnight, obviously. So if you're trying to use this for an area, let's say a place that has a lot of acid mine drainage, just for example, where you have all these metals that are just in this system that are just sitting here that you can probably do something with it. You can put plants there. Like, is it something that like we're gonna try to start this now, and then 20 years from now, see what what we get out of it? Like, I don't know if that makes sense.
Yeah, okay. I can see the organisers hovering, and we don't want to run over.
Yeah.
But maybe we can continue afterwards. Yeah, I'm very happy to speak to anyone and answer that question as well, but I'm going to get dropped down.
And in the sake of time, Liz will be around for the rest of the session and such, so we can have some conversations. Let's thank Liz for her talk today.